Step 1 of DNA replication begins at special sites called the origin of replication. This is where the two DNA strands are separated, opening up a replication bubble. This separating causes something called a replication fork. Here's a closer look at the replication fork, which is right here. The replication fork is basically the Y that forms right about here, that little Y right there, and what creates this unwinding of the double helix of the DNA is this enzyme called helicase. Other enzymes, like the single-strand binding proteins, circled here, help bind to a strand and help stabilize the DNA until it can be used as the template for replication. Another enzyme, sort of a correcting enzyme, that corrects unwinding ahead of the replication fork, it's called topoisomerase. It rejoins the DNA strand and makes sure that the two strands don't overwind. The topoisomerase also moves along the replication fork, so as it's opening, the topoisomerase is going along with the replication fork. The initial nucleotide strand is a short RNA primer, highlighted, circled here. An enzyme called primase starts the RNA chain from scratch and adds the RNA nucleotides one at a time, using the parental DNA as the template. Keep in mind that they can only replicate in the 5' to 3' direction. The actual synthesizing of the new DNA strand is helped by an enzyme called DNA polymerase III. Basically what this enzyme does is takes the nucleotides and attaches them to the parental DNA at the site of the bonding. Once again, you can see that it is adding the nucleotides in the 5' to the 3' direction. Since the double helix unwinds, there will be two strands replicating on each parental strand. This, in turn, will cause a leading strand and a lagging strand. This, once again, the leading strand adds in the 5' to the 3' direction for both of them. On the lagging strand, there's a little bit of complication in what happens and how it replicates. On the lagging strand, after the primase puts down the primer, the DNA polymerase III will start adding the nucleotides in the 5' to 3' direction. As the DNA polymerase III travels down, it will eventually hit the leading strand, and the DNA polymerase will jump back and start from a new primer that the primase lays down and start making more nucleotides and just keep jumping back. In other words, the DNA polymerase III must work in the direction away from the replication fork. The lagging strand is synthesized as a series of segments called these Okazaki fragments. That's why you see all these red little fragments. Then an enzyme called DNA polymerase I replaces the RNA or the red colored nucleotides with DNA nucleotides. Then an enzyme called ligase joins the Okazaki fragments into one long DNA strand. Now let's take an overview of the whole DNA replication process. The strand unwinds by the helicase. The single-stranded binding proteins help stabilize each parental strand. In the leading strand, the DNA polymerase III lays down each nucleotide in the 5' to 3' direction. In the lagging strand, the primase lays down an RNA primer, and the DNA polymerase starts laying down nucleotides in the 5' and 3' direction, and then it jumps back each time to continue the replication process. DNA polymerase I converts these RNA or red nucleotides into the blue or DNA nucleotides. And then finally, DNA ligase joins the little cracks in the new daughter strand, and it connects the little Okazaki fragments into one large original strand. On a side note, the nucleotides that join together always come in pairs. There's a group called the pyrimidines, which are small one-ring structures, and there are purines that are double-ring structures. And all four of them are nucleotides, and the complementary nucleotides are guanine and cytosine, and thymine and adenine. So in other words, whichever ones are on the DNA strand, always, when the new strand is added, the complementary nucleotide is connected.